Biology AQA A-level paper 1 2018. So we are going to go through the questions. We are going to look at the exam tips and the possible answers. So the first question was showing you the chromosomes present in one human cell in mitosis, and the scientists stained and photographed the chromosomes. So the figure 2 then shows that the scientist has arranged the images of those chromosomes in homologous pairs. So that's what we've got. Those are our pictures. And question number 1 is asking you to give 2 pieces of evidence from figure 1. So we're not interested in figure 2. We're looking only at figure 1 that this cell was undergoing mitosis. Explain your answer. So explain, say why. So looking at the picture, try to identify what is the evidence of mitosis and say why using your biological knowledge. So what we can work out from this picture number 1, from figure 1, is the fact that the individual chromosomes are visible because they have condensed. Okay, we can clearly see them. That's the part of mitosis. That's prophase, and then each chromosome is made up of 2 chromatids because DNA has replicated. Yes, that's correct. You can see that on the picture. So going for the next question, we need to tick one box that gives the name of the state of mitosis shown in figure 1. And we've got anaphase, interphase, prophase, telophase. So of course, anaphase, telophase, straight away we disagree. Then interphase, it's not the state of mitosis, it's state of the cell cycle. And of course, chromosomes are visible, so it must be prophase. So tick for prophase. Next, we've got here a question. When preparing the cells for observation, the scientists placed them in a solution that had a slightly higher, so that means less negative, water potential than cytoplasm. This did not cause the cell to burst, but moved the chromosomes further apart in order to reduce the overlapping of the chromosomes when observed with an optical microscope. Suggest, so give a reason why, how this procedure moved the chromosomes apart. Okay, so as we can see here, what I've highlighted, it's less negative, but did not cause cell to burst. So what that means that water moves into the cell by osmosis and the cell gets bigger. Okay, cell gets bigger but does not burst. Right. Question next, the dark stain used on the chromosomes binds more to some areas of the chromosomes than others, giving the chromosomes a stripped appearance. Suggest, so again, give the reason why. One way the structure of the chromosome could differ along its length to result in the stain binding more in some areas. So if they're using the words like differ or different or only one, we need to be thinking, right, so what's different? So what is the difference in here? The difference is found in the base sequence. Okay, and that's the reason why the stain will be bind more to some of the areas. Next question. In figure 2, the chromosomes are arranged in homologous pairs. And what is a homologous pair of chromosomes? So only the definition. So there are 2 chromosomes that carry the same genes. And we know that one comes from mom, of course, so that's a maternal, and the other one is paternal. 1.6 then give 2 ways in which the arrangement of prokaryotic DNA is different from the arrangement of the human DNA in figure 1. So again, you need to go back, okay, and use the figure 1. So what we could see from figure 1, so let's quickly go back to figure 1, okay, that is our figure 1. So let's have a quick look at the arrangement of the chromosomes and come back to the question. Okay, so question, what's the difference? That we know that in prokaryotic cells DNA is circular and it's not associated with histones. Okay. Question number 2. Okay, question number 2. A student investigated the effect of surface area on osmosis in cubes of potato. Okay, so we're looking at osmosis and we've got potatoes and there is a method. So he cut 2 cubes of potato tissue, each with sides of 35 millimeters in length. So that's important information. So he put 1 cube onto a concentrated sucrose solution. He cut the other cube into 8 equal size, so that's important, smaller cubes and put them into a sucrose solution of the same concentration as the solution used for the large cube. So what that says that any mass loss, so any osmosis should be same because should be similar. Okay, because we've got the concentration the same, but it won't be similar because they are different size of the cubes. So we're looking then at the figure number 3, and let's have a look what we've got here. So we've got a mass, we've got time, and we've got the key for different types of the cubes. So what we can see on the graph here is we've got different lines for large cube and different for the 8 ones, and then we've got the mass for those. So let's have a look what the question is asking you. Question is to describe the method the student would have used to obtain the results in figure 3. Start after all the cubes of potato have been cut and consider variables he should have controlled. So if you ask to describe the method, you need to come up with the methods that somebody can repeat step by step. So what would be the method after the potatoes has been cut? So method to make sure that all cut surfaces of 8 cubes are exposed to the sucrose solution. So that's important. We want all of those to be in there. Method of controlling temperature, so could be at room temperature, could be using thermometer. Method of drying cubes before measuring, so that could be, for example, using the blue roll to dry the cubes before you measure the initial mass. And measure mass of cubes at stated time intervals. So you could come up with the fact like you could do it every 5 minutes, but up to every 40 minutes. So anything between 5 to 40 minutes will give you a mark. So now we had a maths question which was quite challenging. So the loss in mass in figure 3, so on the graph that we were looking at, is due to osmosis. So we've mentioned that. So we did say that any mass loss will be because water is lost. With the practical was going on in this concentration of sucrose. So the solution has sucrose. So of course osmosis is taking place from higher water potential to lower water potential, losing water. So the rate of osmosis between 0 and 40 minutes, it's faster in B, and B were those 8 small cubes, than in A, so single large cube. So is the rate of osmosis per millimeter squared per minute different between A and B during this time? And use the calculations to support. So we did mention that will be different because of course the size of the cubes. So coming back to the figure really quickly, let's have a look what they want from us. We're looking at the rate between 0 to 40. So we need to work out the mass, okay, for a cube, large cube, and for each of the small cubes. So we've got that from the graph, and let's have a look what the calculations are like. So firstly, we're looking at the mass loss. So the mass loss between 0, okay, to 40 minutes. So we've got 5.2 for cube A, so that's a single large cube, and 9.2 for a small cube. But then we need to use our imagination, and of course we need to understand how those cubes were cut. So before I did that, I worked out the rate of mass loss. So use the total mass loss divided by 40 minutes. So those are my rates here. And now we're looking at the cubes. So I need to work out the the surfaces of those cubes. So here should be cubed, sorry. So, oh no, it's fine. So what we've got here, the surface, of course, we've got a cube here. So each side multiplied by 6. That's the answer for the first cube. The other cube here, of course, we're cutting into 8. So in total, we will have 3 cuts. So if I'm cutting them into 8, the length, the new length of one side will be 17.5. Do the same calculation as before. So this is the surface of one cube. Of course, we had 8 of them. So multiply by 8. And that's our new answers. So what do we then need to do? We're looking at the final answer. So the rate that I work out divided by the surface of the cube. So 1.77 times 10 to minus 5 it's for the large cube, and 1.56 times 10 to minus 5 it's the rate for the one small cube. So yes, there is a difference. Right, so we're looking at the next question. So you got another graphs here. So bees are flying insects that feed on nectar made in flowers. There are many different species of bee, and scientists investigated how biodiversity of bees varied in different habitats. And they collected bees from 8 sites of each habitat 4 times per year for 3 years. So again, lots of information, lots of numbers here, and the results were presented in figure 4. So what's really interesting when you're looking at those figures? Of course, labels. So mean, okay, number and species richness. We've got the time here, day of the year, and then we've got the habitats, 3 different habitats. So once you read this information, you can then work out what is going on here. So first question, it's asking you what is meant by species richness. So species richness is the measure of the number of different species in the community. So it's just a definition that you need to provide. So next question, what we've got here, it's question number 3.2. So from the data in the figure 4, a student made the following conclusions, and that's a really new typical for AQA. They're giving you conclusions. So number 1 and number 2, highlighted conclusions, and do the data in figure 4 support those conclusions? Explain why. So firstly, using the figure number 4. Secondly, using your biological knowledge, say why. So if here they're asking if they support, so you can come up with the answers for yes and answers for no. Some of the questions will be explaining how they support. So of course, then we're only looking at the positives. So what we remember, we had mean values here. So for the natural habitat, I've given the answer for yes and given one for no. So what's yes? The peak of mean bee numbers in the natural habitats is highest. So we can see that from the graph, and the lowest mean number of bees after 220 days. Okay, so again, we can conclude that here that's the highest, and then 220 around here for the natural habitats. Okay, so then the town is the least favorable for bees. So yes, peak of species richness higher in both natural and farmland, and no, mean species richness is lower in farmland until 125 days. So again, let's have a look at this. So we're looking now at those 2 lines, okay, and yes, they are lower. So make sure for the conclusions like this, you do use the figure. You don't really need any scientific knowledge in here. You're only saying what you can see. So using figure, make sure you use the values as well. Okay, so let's have a look at the next part of this question. So the scientists collected bees using a method that was ethical and allowed them to identify accurately the species to which they belong. And in each case, so just one consideration the scientists had taken into account to make sure the method, okay, so ethical, so reason why was ethical, so of course not to harm the bees and allow them to identify accurately the species to which they belong, so use a key to identify the species. So the same as we had with the graphs was showing you the different habitats, for example. The same idea of using the key should be used. So next question. So just an explain. So again, give the reason why. Two ways in which the scientists could improve the method, okay, to collect the data. So what they could do, they could collect more times because what they say they only done it few times per year. So more points would be present on the graph. And again, anything to do with the diversity, species, richness, of course, remember your index of diversity formula. You cannot work out if you've got only mean values or the species richness. We need the values for the individual species. So count number of individual species so that they could calculate index of diversity would be another answer, right? So here, three of the bee species collected in the farmland areas were those different. They had those different species, and what do those names suggest? So again, give the reason about the evolutionary relationship. Any question about that? Think about being closely related, okay? So common ancestor between those bee species and explain your answer. So use your knowledge, okay? So two of those species, okay, are more closely related because they are in the same genus, okay? So how can we work it out? Look at the names, Andrena and Angena. Here we have different ones. So this is the genus, this is the species. They are closely related looking at the name, binomial naming, okay? So that's where the answer is coming from. Right. Looking at the question number 4, they're asking you about enzymes. And enzymes is a topic that you cannot escape from. Every single exam will ask you a question about enzymes and proteins. So what they want here is the formation of an enzyme substrate complex increases the rate of reaction. Explain how. So again, use your biological knowledge. Say why. Why does that take place? Because they reduce activation energy, and of course, without enzyme, very few substrates have sufficient energy for the reaction. So 2 marks for and then the scientists measure the rate of removal of amino acids from a polypeptide with and without enzyme present. So with the enzyme present, 578 amino acids were released per second. Without, 3 times 10 to minus 9 were released. So of course here there is a huge difference between them because we know if the enzyme is present, the reaction will go faster. When we explained in the question before why. So calculate how many times the rate of reaction is greater with the enzyme present and give your answer in standard form. So that's important. Use the standard form. So with, I've said it's 578 per second. Without, again, I've said what it was. So what my calculation is here, I've just divided those values, okay, and got the answer of 1.93 times 10 to power 11. And that's how many times it's faster. Okay. So next we've got the investigation, an enzyme that catalyzes the following reaction, and this is the hydrolysis of ATP. And the scientists set up 2 experiments, C and L. So C is with the enzyme with different concentrations of ATP. L is exactly the same with enzyme, different concentrations of ATP, but what's different? They've got a sugar called lactose. So the scientists measured the rate of reaction in each experiment, and the results are shown as here. So what I've done here, Okay, of course, comparing those, so L and C, we can see which one is faster, which one is lower. The maximum rate of reaction is 1.4, and what we've got on our axis is the rate of reaction against the concentration of ATP. So what we're doing here next, looking at the question, and they're asking you to calculate the rate of reaction of the enzyme activity with no lysose at 2.5, okay, of ATP as a percentage of the maximum rate shown with lysose. So again, where are the calculations coming from? So you've got the maximum rate with lysose, which is 1.4, and no lysose at 2.5, it's not 0.44. Okay, so to show you where is this coming from, this is 2.4, 2.5 the concentration. Draw the line up for C. Okay, the value is not 0.44. So again, our calculation is straightforward. There's a percentage, so not 0.44 divided by 1.4 times 100, and the answer is 31.4%. So that's the maximum. So that's the percentage, sorry. Right, next question. Again, enzymes. So lactose binds to the enzyme. Okay, so means it's a substrate, and suggests, so give the reason why and how. A reason for a difference in the results shown in figure 5 with and without lactose sugar. So again, you're using the figure, and again, to remind you, what was the figure? Was the difference in the rate of reaction. Right, so using your biological knowledge, you should then recall all the keywords about the enzyme. So binding alerts the tertiary structure of the enzyme. So this causes active shape to change, active site to change shape. So more enzyme substrate complexes can form per minute. So other words, you here using your knowledge about the induced fit model to explain the enzyme's action. So let's have a look at the next question. Draw a structure of amino acids. Straightforward, hopefully. For everybody, we've got that. Table number 1 then shows you mRNA codons and the amino acids coded for by each of the codons. So remember, codon, it's a triplet on mRNA. And it also shows some properties of R groups. So where we've got the properties of R groups, they are here. Okay, so we're talking about not charge, positive charge, and negative charge. The question here is asking you what it's meant by degenerate genetic code, and use example from table 1 to illustrate your answer. So you know that the second marking point is coming from just using the table. So the answers definition: more than one codon codes for a single amino acid, and then using the example from the table. So I've got, for example, val amino acids, and those 4 different codons coding for it. So let's have a look, where is this coming from? So where we've got val is here. So how do you use the table? G, U, U. It's the first codon. Then we've got G, U, C, another codon. G, U, A, another codon coding for val, and GUG. So all of those are coding for val. So straightforward, make sure you know how to use the codon table. And questions here: scientists investigated changes in the amino acid sequence of a human enzyme resulting from mutations, and all these amino acid changes results from single base substitution mutation. And the enzyme, it's made of 465 amino acids. What we need to remember, that one amino acid, it's coded by 3 bases. Okay, and we've got a table here, shows you different number of the amino acids. So what is the minimum number of bases coding for this polypeptide? So we've said one amino acid coded by 3 bases. So multiply this 465 by 3, and we've got 1395 for the answer. Right? Next question is to use information from table 1 to tick one box that shows a single base substitution mutation in DNA that will result in a change from val to ala at amino acid number 203. So looking at the mark scheme, okay, the mark scheme gives you the first answer, but it's incorrect because well, first answer is the single base substitution. Yes, it is, but it does not change val to ala, change different amino acids. Second option, then it does change val to ala, but as you can see, it's not a single base substitution mutation because it's uracil exchanged by cytosine and then uracil by adenine. So it's obviously it's not a single base substitution. So there was a mistake in that question. So next we've got a change from glue to lys at amino acid 300 had no effect. No effect, that means that the same amino acid will be coded for due to, of course, we know degenerate genetic code on the rate of reaction catalyzed by the enzyme. So the same change at amino acid 279 significantly reduced the rates. That means that a different amino acid will be produced. So use all the information and your knowledge of protein structure. Okay, so information, what are the yellow highlighted bits, and knowledge of protein structure to suggest, to give the reason why the difference between the effects of those two changes. So again, what we can do, we're using the information. So information, looking at the key from the table, charges, and then we're using the information about the numbers here and the structure of the protein. So both negatively charged to positively charged change in amino acid. So that's from the charges, and change at amino acid 300 does not change the shape of the active site because of course it's the same amino acid that been coded for using the fact of degenerate genetic code. And amino acid 279 might have been involved in the hydrogen bond, so the shape of the active site changes. Okay, and let's have a look at question 6. So we've got a faulty form of meiosis. Okay, so what we've got here, of course, meiosis information, we've got that it's a diploid parent. So we've got 2n first division. So it shows you the content of the cells of the first division, and the second division, it shows you that rather than getting four haploid cells, we are going to get only 2 cells, and both of them are diploid. Okay, so what we can then look, of course, it's a question. So complete figure 7 to show the chromosome content of the cell that would result from a normal mitotic division. Okay, so we did say that the first division, of course, in the after first division, we are going to get 2 diploid cells because that's what meiosis is. And after second division, we are going to get 4 haploid cells. So your drawing should look like this. Right. Next question. We've got a table that gives you information about the observed and expected values, and we've got lots of information here. So I let you have a look through those, but the yellow bits are highlighted and they are really important. Okay, so using this information, the scientists used the following null hypothesis. So the proportion of plants that produce diploid gametes will not change. Okay, so look, the proportion of those, so if you got 4 here, that means in the next generation you're going to get 4 as well. They stay the same, they don't change. So complete table 3 to show the expected number of plants that did not produce 2n gametes, so diploid gametes, and the expected number of plants that did produce 2n gametes. Okay, so I'm sorry here, my labels are incorrect. So did produce 2N, okay, that means that they stay the same and did not produce 2N, there will be different. Sorry, so the labels are just all the way round. So how we can approach this, we can work out the total of the plants that we're going to get here. So what we're going to do, we're going to add them together. So 14 and 42 gives you 56. Okay, so 56 is the total that we can play with, and we need to use that to work out the expected values. So we did work out that the ones that produce 2N are going to stay the same. So we can take it away. So 56 minus 4, because they won't change, gives you 52. And this is how we're going to complete our table. So the one that did not produce 52, the one that produced 4. Right. So then the scientists used the null hypothesis and the chi-square test. And after one cycle, they calculated the chi-square value was 350, but the critical value was 3.841. So what we can see here, that the chi-square is higher than critical value. In this situation, we are going to reject the null hypothesis. Why? Because there is less than 5% probability that the difference between observed and expected occurred by chance, and the calculated value, what we've said, it's higher than critical value, so null hypothesis is rejected. Right. So using your knowledge of directional selection, explain, so say why the results shown in table 3. Okay, so what we've seen in table 3, it's the fact that the scientists used for breeding plants that produce diploid gametes. So those plants pass on the alleles for production of diploid gametes to the next generation. So the frequency of the alleles for production of diploid gametes increased in the population. Okay, so this is what we can conclude about the directional selection. Question 7. Person is bitten by a snake and the snake injects toxin. Okay, antivenom is injected as treatment, and antivenom contains antibodies against the snake toxin. So in other words, the snake toxin works as antigen. And this treatment is an example of passive immunity. So passive immunity is the form when the antibody is injected. So explain, say why, how the treatment of this antivenom works, and how it is essential to use passive immunity rather than active immunity. So in other words, why passive, okay, would be better. So antivenom antibodies bind to the antigen, and it causes its deconstruction, because that's what the function of antibodies is, causing deconstruction of antigen. And active immunity would be too slow. Why would be too slow? Because they will produce their own antibodies, while here you're just injecting in the passive immunity, right? So next question. We've got a figure that shows a procedure used to produce antivenom. So antivenom is the one that contains antibodies. So you've got the snake, collection of the venom, so mixture, selection of the animals, preparation to use for the vaccine. What's vaccine? What's the main purpose of a vaccine? To of course release antibodies and produce lots and lots of memory cells, which will act faster because they're going to produce more and faster antibody. Okay, so and then here we've got the vaccination. And then we've got the what we wanted, with antibodies, which will be found in the blood plasma. So the mixture of venoms from several, so that means different snakes on the same species, is used, and suggest why, so use the reason. So in other words, you need to think, right, several snakes means different snakes. So what was to do with the snake? What's the venoms? There are antigens. So the reason, because they might be different forms of antigens, and different antibodies would be then needed in this antivenom, right? 7.3, then another maths. So we've got the horses and rabbits that were used to produce antivenom, and you've got information here to use and give the reason which animal would be better for the production of antivenoms, and use a calculation to support your answer. So of course, what we're looking here, it's the our numbers. Okay, so where those answers coming from the mass of the specific animal, and the amount of the antibodies collected. So what we did here is 350 divided by 13, and then 2 multiplied by 13, and then 2 multiplied by 13. So look, doing that calculation, we can see clearly that horses will have more antivenom, so more antibodies could be collected. Then we've got during this experiment shown in figure 8, so you must use figure 8, the animals are undergoing observation by a vet. And why would that happen? Of course, what is injected, they've given them the vaccine, so to make sure that they don't suffer from the vaccine. So that's the reason. Right, next question. During vaccination, so again, write down everything what you know. What's the key word? So B says antibodies, memory says faster. Each animal is injected with a small volume of venom. Remember, venom was antigen, and 2 weeks later, so that means it's a second time, is injected with a higher volume of venom. So use your knowledge of humoral immune response, so that's to do with B cells, to explain, use your scientific knowledge, this vaccination program. Okay, so all of those things that we just write around the question are important. So what we need to know, that B cells are specific to the venom produced by mitosis. B cells produce plasma cells and memory cells, and the second dose produces antibodies in higher concentration and quickly. Okay, so that's just a model answer that you already know, right? So again, we've got here a treatment, heat treatment on mass transport, okay, in plants. So they've given you lots of information here. So they applied steam to one short section of the leaf. Then you've got this in the figure 9, and they did not apply steam to the leaves of control plants, and then they supply carbon dioxide. Okay, so carbon dioxide, it's to do, of course, with the carbon, so it's to do with the carbohydrates. So that will be then the transport in phloem. Okay, and here they recorded the water content, okay, and then they were looking at the carbon. So anything to do with the water, it's xylem. Anything to do with carbon, it's then phloem. So we've got control group, and then we've got the heat treated plant, 0 hours, 4 hours, okay, and then we've got the table that shows you the water content of leaf. Okay, so water content of leaf, difference between A and B, but also you've got the standard deviation. Any question, any table graph with standard deviation means they want you to use this information. So in here we've got the fact that they overlap. If they overlap, that means there is no significant difference between those. So looking at the question, the scientists conclude that this heat treatment damaged the phloem. So that's the conclusion. Okay, so that means that there is no movement of this carbon. We discussed carbon, it's to do with the phloem, and explain how. So using your biological knowledge, the results in figure 9 support this conclusion. So you here write nothing else than about this conclusion. So what we could here write is lots of things, okay, but movement in phloem requires living cells, requires ATP, and the heat treatment damages living cells, so transport in the phloem throughout the plant, only in the plants that were untreated, so the control p value. experiment. And then they concluded another statement here that this heat treatment did not affect the xylem. So, other words, that means the water should be moving fine, and explain the results of that using the table 4. So have a look at the table 4 again, and the water content of the leaves was not different because the standard deviation overlap. So that's why I've said you must always use the standard deviation, and the water is still being transported in the xylem to the leaf. Okay, so that's using only table 4. That's how we can explain our results. Next. So here we've got another investigation, the movement of the ions from the soil. So from the soil, that means we're looking at the xylem. And then we've got the information that they use the young leaves of the heat-treated plants and 2 leaves of plants that were not heat-treated. So the control and heat treatment was applied halfway up the leaves. So halfway up the leaves, that means that this ions will not get to the leaves, okay, if that, or they will get or they will not. So it's something to do with the transport from the roots to the leaves. And the scientists determine the concentration of ions in the top and lower halves of the leaves. So this is what we've got. We've got old and young leaves, we've got the ratio of the concentration, we've got heated and unheated plants. Okay, so from them we can see that, for example, in the young leaves, the heat treatment halfway through the leaf, okay, had the results really low for the concentration in top half of the leaf. Okay, so what we can then see, it's a question, what can you conclude? So what you can conclude is then you, using the information, using your knowledge, okay, text and the graph about the movement of ions. Okay, so what you can conclude, we don't have any statement, it's up to you now. So heat treatment has a higher effect on young leaves, so that's what we've seen, than old. Heat treatment damages the phloem. Okay, ions moves up the leaf, so if they move up the leaf, they need to use, of course, xylem, so the ions is transported by the xylem in all the leaf. Right, and that is, you could use many more options here, but that's the most common. Right, question number 9. Describe, to say what you can see, the role of 2 named enzymes in the process of semi-conservative replication of DNA. So of course we've got 2 enzymes, name them, and the job is to talk about the bonds. So helicase causes breaking of hydrogen bonds between DNA bases. Polymerase joins the DNA nucleotides, forming phosphodiester bond. Right, then we've got question number 9. Again, another investigation, the function of the eukaryotic cell protein called cyclin A, and this protein is thought to be involved with the binding of the enzymes required at the start of DNA replication, and we've got different ways here. So C, control, B, antibody, F, RNA to prevent translation, and F, prevents translation, and added a protein, cyclin A protein. So what is the job of it? It's needed at the start of replication, which we can work out from the stem of question. So they work out the percentage of the cells in each culture in which DNA was replicating. We've got results in table 5. Okay, so different results, as we can see here, the pattern 91, 92, 11, 10. Okay, so what's the question? So this explanation for those results. So other words, why are they high? Why are those low? Okay, so using your biological knowledge, so treatment, the antibody binds to cyclin A, so it can't bind to DNA to initiate DNA replication. That's what we say, it's needed to start. Treatment E, then RNA interferes with mRNA formation, and in treatment F, added cyclin A can bind to DNA to initiate DNA replication. So hence we've got that high number. Finally, question 10, describe, so again, say what you can see, the gross structure of the human gas exchange system and how we breathe in and out. So here they're actually asking you for two things, breathing in and out, and then gross structure. So 6 marks for this, really easy question. So where the marks coming from? So the marks coming from, of course, the gross structure. So you could list that in the right order. So trachea, bronchi, bronchioles, and alveoli. And then how do we breathe in and out? So breathing in, marks, breathing out, marks. So breathing in, diaphragm and external intercostal muscle contract. What does that do? Because what we're doing, we're saying how, so causes volume increase and pressure decrease in thoracic cavity. Breathing out, diaphragm relaxes and internal intercostal muscles contract, so causes volume to decrease and pressure increase in thoracic cavity. So really nice friendly question here at the end of the paper. Then we've got the mucus produced by epithelial cells in the human gas exchange system contains triglycerides and phospholipids. So compare and contrast. So both aspects here you need to say, include in your sentences both of them, triglycerides and phospholipids. Structure and properties. So again, pay attention. Marks for each of the aspects, structure and properties. So both of them, there are plenty of things you could talk about. So contain glycerol, contain ester bond. Property, it's the fact that insoluble in water. So structure, fatty acids on both might be saturated or unsaturated. They contain elements of carbon, hydrogen, and oxygen. But phospholipids also contain phosphate, okay? And fact about the triglycerides having 3 fatty acids and phospholipids has 2 fatty acids plus phosphate group. So 1 fatty acid was replaced by a phosphate group. So that's the difference, okay? And the last question here, we've got the mucus also contains glycoproteins. One of those glycoprotein is a polypeptide with the sugar lactose attached. Describe, so again, say what you can see, how lactose is formed, so the process, and where in the cell, specifically in the cell, it could attach to the polypeptides to form glycoprotein. So we know that Golgi body is involved in that process. So how to do it? Looking for marks, start with the easy mark and then work it down. So what? Looking at the question. So firstly, what is the lactose? It's glucose and galactose. So it's the disaccharide joined by condensation reaction, joined by glycosidic bond, and added to polypeptide in Golgi apparatus, Golgi body. So that's the end of 2018 paper 1. So be careful, read the questions properly, okay? As you can see, paper was not that bad, okay? The wording is the one that you need to pay attention to. Spend time labeling the question, looking at the comment words. It's better to not to finish the paper but get the marks. Remember, each mark is a mark, okay? There is no weight to the marks. Just be calm, look at the questions properly and make sure you know what you're doing. So that's everything for paper 1. See you later.